So if I can make a lot of copies of this gene, then I can make a lot of copies of this protein, right? And if I can make a lot of copies of insulin, I could use insulin in the medical supply world. So that's how we're using bacteria, for example, to benefit us. So the central dogma is the same in every organism. If it wasn't, I wouldn't be able to take that insulin gene and put it into a bacterium, right? So every organism is going to go through that whole transcription translation process, which means you can really put any gene into any organism and have it translated. This allows us to clone genes as well as modify genes. So maybe you have a gene that isn't functioning properly. You could take out the pieces that don't work and put in new nucleotides that are needed, right? So maybe there's a mutation and we can fix that mutation. We do this quite often with bacteria and plants. In the plant world, we think of agriculture, making plants more like weather resistant or pesticide resistant, or able to be their own pesticide to get rid of pests. So we're improving characteristics of one sort or another, and we use our DNA, which is recombinant DNA. So recombinant DNA is really two DNAs from two different sources. So quite often we're talking about human and bacterial. We might be talking about bacterial and plant. We might be doing bacterial and mice, or viruses even. So we need a vector. Often that vector comes in the form of a plasmid, which is what we're going to talk about today. It sometimes comes in the form of a virus. So here comes the bacteria, because the bacteria are easy to work with. They are opportunistic, which means they're taking in DNA right through their cell walls, and they're able to incorporate DNA from their environment into their own genome. Okay, so we have surface transport proteins that are specialized for the uptake of naked DNA. So it's taking DNA from the environment and bringing them into the bacteria itself. They're going to incorporate it into their own genome. Often they're going to incorporate it into a plasmid, which is an accessory DNA. It's an extra piece of DNA. So we know that we can do this because we saw it happen in Griffith, right? Remember the experiment with, he was trying to find the vaccine for pneumonia, and he worked with the R and S strain bacteria, and he found that the R strain, which was not virulent, was transformed into the S strain, which was. So that's where we got the idea that we can actually change the genome of an organism in order to change their phenotype. So we're going to incorporate DNA bits into the chromosome of the bacteria, and this is going to cause the bacteria to express new genes, or whatever organism you're incorporating into, okay? So this forms what we call recombination, which is just DNA from two different sources. So we haven't had a lot of background on bacteria. We do talk about them, and we take for granted that you know a lot of things. You know that they're single-celled prokaryotes. We can trace them back. So the next piece we go into is evolution. We can trace fossils back 3.5 billion years ago, which means that this whole gene technology was, or like how genes work, right? This has been something that has been highly conserved over evolutionary time, because it still is how we transcribe and translate genes. We know they reproduce through binary fission, meaning they're going to make copies of their DNA and they're going to split in half, which allows them to reproduce very quickly, about every 20 minutes. It's exponential growth. You should be able to do the math on that, which means we can make 100 million colonies overnight. So if there is a gene that I want to express, bacteria are the perfect lab organism. If I want to manufacture a protein in the lab, bacteria are perfect for that. They're incredibly diverse. We can find them everywhere, right? We can find them in the soil, we can find them in the water, we can find them in the air, we can find them in volcanic vents. We find them everywhere. So a little bit about their structure. The capsule is has a cell wall. So the capsule here the cell wall is made out of polysaccharides, which is a glycocalyx, is what you would refer to, polysaccharides. The cell wall is mostly made up of peptidoglycan. So this is taking us all the way back to chapter 3, I think, when we did organic molecules, right? So that peptidoglycan allows it to maintain its shape. This component of the cell wall is what differentiates a monarin, a eubacteria, from a archaebacteria. It has a plasma membrane surrounded by surrounding the cytoplasm, and that's going to regulate material moving into and out of the cell the same as it does with us. Lots of inward folds. Because it's a small organism, it needs to have a great efficiency. And we know that folds increase surface area, which increases efficiency, right? Ribosomes for protein synthesis. I just looked at the clock. I might need to talk a little bit faster. This is getting into the plasmids here. See the normal DNA of the bacteria. This is the chromosome. Then you have these little circular DNAs. They're called plasmids. They're accessories. These are genes they pick up from their environment. Okay, so this is what we're going to take advantage of when it comes to recombinant DNA. I'm going to kind of skip over the structure right now because I want to get to the plasmids. So the plasmid may contain superpowers like antibiotic resistance. So we know that a lot of the bacteria have evolved over time, right? And we have a hard time fighting them now because they have become resistant to our antibiotics. Every time we introduce a new antibiotic, some of the organisms, some of the bacteria will have mutations that allow them to be resistant to that bacteria. They survive, they pass on their trait, and the population changes to a resistant population, right? That's evolution. And it has happened with the introduction with every single antibiotic we have introduced. Okay, so that's one of the features we're going to take advantage of when we do gene selection. So the plasmid, the circular DNA, it replicates independently of the bacterial DNA. So it's not necessarily replicating with the chromosome genome. It is replicating on its own, kind of like mitochondrial DNA replicates on its own. Okay, so we can take those out and we can introduce them into other organisms. They might pick up DNA from the environment that gets incorporated into their plasmid, which is what creates the recombination and the great diversity of bacteria. So we can use this plasmid as a vector. A vector is a source we use to move the DNA from one organism to another. So if I incorporate a gene of interest like insulin into this plasmid, I can then move that plasmid into other organisms. Sometimes we use viruses as well, but today we're talking about the plasmid. We need a restriction enzyme in order to cut this plasmid in order to be able to fit a new piece of DNA in. So restriction enzymes are found in bacteria. They're actually like their immune system. The restriction enzymes are used to restrict the life of a virus. So they're going to cut up viral DNA. They get incorporated, kind of like how we make different antibodies to match different antigens that we're presented with. Okay, so then they recognize that genome, they recognize that sequence, and they're able to go after that sequence when they see it. Okay, so restriction enzymes we use in the lab to cleave or cut plasmids and other DNAs. We also need the enzyme ligase. Ligase, you'll remember, seals the gap, right? We talked about that with the lagging strand of DNA. So, these, knowing how DNA replicates, we're able to take pieces and parts of that process and use that in this recombination technology. So we use the restriction enzyme to cut it and the ligase to glue it back together. So our restriction enzymes are also called endonucleases. You'll see it called either way. We're going to work more with restriction enzymes. They have names. Usually the first part tells you what organism it comes from. So E. coli is a very common one, refers to E. coli. Our restriction enzyme this was the first one that was discovered. So they have numerals, and that is like in the order of their discovery. So we work with ECO R1, R2, R3, and they each find a specific sequence of DNA. So ECO R1 is specifically looking for the sequence TTAA. So when it reads TTAA, it will cleave after that A. And remember, we read 3 to 5. So TTAA, it will cleave after that A. So that's where the restriction enzyme makes its cut. You can see here. Notice this is a jagged cut. We call that sticky ends. You need sticky ends if you're going to be able to seal two pieces of DNA together. So if we use the same restriction enzyme on two different DNA strands, one from the bacteria and one that is from our organism of interest, we use the same cleavage enzyme, we're going to create the same cut, right? So that means they both have an AATT that's hanging over, and that is complementary to the TTAA that is hanging over. They make use of palindromes often too. So complementary strands are able then to stick together. So we're able to use that same one, cleave the insulin gene, AATTs hanging out there. It's going to bind with TTAA, and that's how we incorporate it into the bacterium. So what you're going to view tonight is your short little video on CRISPR. So CRISPR has been, I don't know if I can say new anymore, but it was new in the last handful of years. It still seems new. You actually see commercials referring to using CRISPR to take care of cancer, although they don't use the word CRISPR, but some of the medicines that they're referring to, it's using CRISPR. Anyways, this is revolutionary biotechnology because we can use it like molecular scissors to take out damaged DNA and to then insert functioning DNA. So this allows us to fix genetic defects, and that's what you're going to be reading about. They call it gene editing, not reading, you're just watching a video. So the ligase we know is going to seal the gap, right? So we're back to cleaving restriction enzymes. Here's our TTAA from ECO R1. We make this in the E. coli that we're using. We're going to make the same cut in our gene of interest, like insulin. Now the insulin has AATT overhang, TTAA overhang from the plasmid. These two will fit together. So ligase then seals the gap between the ECO G and the insulin A, for example. So when we're able to get DNA to uptake plasmids, it's called competency. So we need that word when we do our transformation lab. So if a bacteria is able to take up DNA, the bacteria is competent. And we'll talk later about how we make them competent. So then the recombinant plasmid is taken up by the bacteria, and the bacteria makes more and more of that plasmid. It replicates very quickly, and the bacteria makes the protein that we want. Okay, so we're going to make lots and lots of insulin now. So we need a way to select those that have the gene and those that don't have the gene. So because we only want to grow the ones that have the gene, right? So we call this selection. We need a way to select the colonies that we want. And so we are going to put in a selective pressure. When we say selective, we're talking about natural selection, right? Survival of the fittest. Those that have the trait survive, those that don't. Okay, so we're going to use antibiotic resistance. So if they're resistant to penicillin, for example, we're going to use penicillin on our growth plates. Those that have taken up the plasmid that allows them to be resistant to penicillin and therefore make insulin are going to survive. Those that didn't take the plasmid up and therefore aren't resistant and can't make the insulin, they're not going to survive. Okay, so we're going to put both of those traits into one plasmid. we're going to put the insulin gene as well as the AMP resistant, penicillin resistant is the example I'm using, gene in there. So if they picked up the penicillin resistant gene, they also picked up the insulin gene. And if they didn't, they didn't. In the lab we're working with, we use ampicillin. So that's why AMP slipped out. We would use an ampicillin resistant gene in our plasmid. So those that survived are the ones that can express the gene of interest, insulin, for example. Okay. So you can imagine what we would use, like it's not enough just to be able to do it, right? We have to have a purpose for doing it. So we make genetically modified organisms, which means their genes, their genome has been altered. It allows them to produce different proteins than they could before. Often it's used in agriculture to produce crops that will resist insects, resist weather. Corn, cotton, potatoes, bananas are very common examples. They produce a bacterial toxin that will kill pests. We're able to make weather resistant organisms. We take genes from, we have these anti-freezing genes from flounder in like the deep sea ocean where it's very, very cold, right? And we've moved them into fish and strawberries, and we call them fish berries. And so they're able to resist the cold weather. So it extends their growing season. We've improved the quality of the rice that we grow by inserting a gene that allows them to produce vitamin A. So we're able to alter the benefits that we get out of the organism. So we can see where vitamin A is deficient, right? So if we then grow that rice, which can, like rice is easy to grow and it grows in a large quantity, correct? So if this is an area that typically has vitamin A deficiency, by incorporating the vitamin A into that rice, we're helping that population. We can also use it for bioremediation. So we have manufactured bacteria that can actually digest hydrocarbons. And so they're able to get rid of oil spills. Like they take in the oil and they digest it. They can remove oil, they can remove pesticides, fertilizers, and some other toxins from the environment. So this is showing you one area that has gone through that bioremediation where we've used those bacteria that clean up the environment. And you can see how that water has changed in its transparency, for example. Okay. So we also use it in medicine, and I kept using the example of the insulin. So we can call that a gene of interest by incorporating it or any other gene that we need to make for people in the medicine world. We can make more and more of that protein. And I'm going to skip the, do you know your bacterias? Bacteria are named according to their shape and according to how many there are. So like a rod shape is a bacillus, Staphylococcus bacillus, for example. Circles are called coccus. So like Streptococcus pneumonia, strep throat. If you have two, it's a diploid. If you have a pile, it's a staph. You have Spirillus as well. And then this one is the Vibrio. So this just helps you. Bacteria are typically named according to their shapes and their community or lifestyle, if you will. So that helps us know something about the bacteria just by knowing its name. And I'm going to skip the labeling because we were a little bit shorter on time. So hopefully we got the things that we need for making recombinant DNA, right? We need a vector, we need a restriction enzyme, and we need a ligase, okay? We're going to work more with restriction enzymes. We're going to work more with plasmids. And today you're going to explore a little bit about the CRISPR gene, okay? And answer the questions that are included in that. So we got 5 minutes. I was just a little bit worried there for a minute that I might not get through it all. What questions do you have for me?